Hey, find the lecture. Okay, chat box is open. Last time it said drug lecture 1 of 2, and this one's 2 of 3. Maybe we'll just go on indefinitely. I've forgotten the last one. We're going to also talk about ibuprofen at the end, which is another form of pain management and dissociation from the sensation of participation and the sensation of physiological status, where you're sort of disrupting the body's acknowledgement of a physiological state. So we're done talking about alcohol. I'll put all of the alcohol stuff back in for right at the end of the semester, as we're getting ready for that last test. I'll throw the alcohol stuff back in and we'll chat about it and go through it all again. But this is our departure slide. We've known about hangovers for a long time. We have known about alcohol for a much longer, with the exact same amount of time, I guess, we've known about hangovers. As soon as something is introduced, it is human nature to exploit it and be penalized by that exploitation. That just seems to be what we do as a species. And uh, but we've learned more and more about fermentation and the distillation after that, and the physiological mechanisms of hangovers, all that stuff we're still learning, but we have a pretty good understanding. There's a pretty good understanding of what alcohol does both acutely and chronically. And a lot of the mTOR signaling with alcohol is being identified now. There's a picture that's coming. If you've ever watched Bob Ross paint, he doesn't paint like, okay, let me do the mountain start to finish, totally detailed. The rest of the canvas is white, and the mountain it looks like you could scale it. And then, okay, next is the tree. He doesn't do that. It develops. The whole picture develops as he goes. And with alcohol, we're developing the picture. There's a hazy mountain there. There's a hazy landscape of trees and a lake and whatever. And the happy clouds and squirrels and stuff. But it isn't photographic detail with alcohol. As we get into THC and some of the other drugs as well, but CBD, THC, as we get into cannabis, the landscape isn't even really there. We just know that there's a landscape. We know that a landscape exists with mTOR metabolism, and we're pretty good at understanding what causes the munchies. We figured that much out with cannabis, but the rest of it is a little bit hazy, and we talked about that, the contradiction of mTOR with CBD on whatever it was, Wednesday. And we'll talk about the same thing with THC today, and we'll finish probably early. But just remember that what people say about drugs and truth are usually pretty different. What people say about things like, there's LSD, there's shrooms, right? There's tobacco, there's alcohol. Like, what people say about these things usually contradicts science and evidence and experimentation and the results and dissemination of all sort of the known sciences. So making sure we stay in a very as level-headed and evidence-based as we can be in all this. But we'll talk about cannabis, which has so little understanding of the metabolism. There's increasing understanding of, oh, hey, look at that. Oh, hey, look at that. Oh, hey, look at that, in its interactions and in how it works. But the understanding of its acute and chronic metabolic reactions in the body is at least hazy, if not something more foggy than that. So remember this, there's TrkA, tropomyosin receptor kinase A, tyrosine receptor kinase A, whatever you want to call it. That's where nerve growth factor is going to bind. But remember TrkB. TrkB is where BDNF, brain-derived neurotrophic factor, will bind. And so there are different tracks, there are the cell surface receptors, and factors bind to them. A factor is a protein. Factors are protein, insulin-like growth factor, atrial natriuretic factor, or usually it's called peptide, ANP, but ANF, it's often called too, atrial natriuretic factor, just a bunch of proteins, right? You see bradykinins over here. Down here, we see prostaglandins. Over here, this would be an AMPA receptor. Let's let some sodium in. But down here, we see anandamide, that AEA, anandamide. That was the first ever endogenous within, as opposed to exogenous without, outside, right? Endogenous within, the first ever cannabinoid, endogenous cannabinoid discovered was anandamide. And it's made in part, not in full, but it's made in part by arachidonic acid. Right? Prostaglandins over here, that's made in full by arachidonic acid. Anandamide is made in part by arachidonic acid. And we're seeing in this one an opposite effect on Adenolith Cyclase. There's an opposite effect that they're having on Adenolith Cyclase. And it appears that cannabinoids and opiates or opioids, remember there's the natural and the unnatural synthetic version. Opioid, that's your synthetic version. It appears that there's acting on the same receptor here. That's not true. They have their own receptors, but in this case, we're looking at an opposing effect from prostaglandins, which are activating adenolith cyclase. Now, adenolith cyclase converts ATP into cyclic AMP. Cyclic AMP binds to pKa, activates pKa, and it depends where you are. Are you in a nerve? Are you in the liver? Are you in a fat cell? Where are you in the body? pKa is going to have some different effects. And the first ever identified effect of the cannabinoids was this same adenolith cyclase signal, and which is how the opioids exert their primary action, is right there. Now, remember this diagram. There's tons of stuff, tons of cell surface receptors. This isn't even close to all of them. There are so many different cell surface receptors, and some of this stuff you'll recognize. You see the prostaglandins up here. You see prostaglandins down here. You see prostaglandins over here. That's prostaglandins over there. There's different kinds of prostaglandins. It's a fatty acid of prostaglandins. Remember, this comes from arachidonic acid. If this is your phospholipid bilayer right here, this gray thing inside of it is a bunch of cholesterol, a bunch of fatty acids, and arachidonic acid is one of them. And PLA2, phospholipase A2, will extract it, right? It will release that arachidonic acid, and cyclooxygenase converts it to prostaglandins. And it just depends. The different forms of prostaglandins, where it's going to bind, what kind of receptor. Everything in gray over here, you can ignore. An AMPA receptor, an ionotropic receptor, as compared to a metabotropic receptor. When we're talking about mTOR signaling and any type of signaling, we have a signaling cascade. We're seeing the same stuff down here. There's your adenolith or Denylol cyclase, two words for the same thing, adenolith or Denylol, same thing. ATP is converted into cyclic AMP. Cyclic AMP binds to pKa. pKa has its actions. And we're looking at a metabotropic receptor for these signaling cascades. An ionotropic receptor is, okay, ion, come on in. That's an ionotropic receptor. And so the AMPAs, let's let in some sodium, NMDA, let's let in sodium, and let's let in some calcium at the same time. but bradykinins, histamine, right? So if you take your antihistamine, you take your Benadryl, right, you're stopping the release of histamine, or it's like an inverse agonist of the action of histamine. And so you are impairing or inhibiting or obstructing the action of these things. There's drugs for everything. Everything has a drug for it. But prostaglandins, just remember that one. Eicosanoids are just 20 carbon fatty acid things, stuff that's made almost entirely from arachidonic acid. Now, the opioid receptors, just looking at the bottom down here, there's 4 major kinds of receptors. There's 3 here, but whatever, you see the 4 over here, and there's 3 of them. And their primary action is going to be through the signaling cascade. Their primary action, how they are going to induce that state of loopy pain relief, where I wrote it up in this little section right here. So Adenolith Cyclase, that's down here, right? This is an enzyme, an ACE, Adenolith Cyclase, that converts ATP into cyclic AMP, right? That cyclic AMP is going to bind to Pka. Pka phosphorylates tons of stuff. Again, like if you're in the liver, maybe it's phosphorylating phosphorylase kinase, which then phosphorylates glycogen phosphorylase, and now you're mobilizing carbs. If you're in fat, if you're in a fat cell, pKa is going to be phosphorylating hormone-sensitive lipase and perilipin, and you do those 2 things, and that allows you to mobilize your fat. Here, right, we're in a neuron. Here, it's this CREB, the CREB. That's a transcriptional factor. And that is going to bind to a little segment of DNA, and transcription will be affected. Now, you remember transcription and translation. Transcription is nucleic. This is, let's make some mRNA. Let's make a little recipe. We'll send that recipe to the ribosome, to the kitchen, to the ribosome to cook up a protein. So that would be translation, will be at the ribosome. mTOR is turning on translation. We're talking transcription. We're talking pre-mTOR stuff. And when we're activating this thing, there's some nice articles about this that say the cyclic AMP PKA CREB pathway contributes to the analgesic effect of analgesic, this non-pain effect. But it's also memory formation, as we discovered in previous study. Phosphorylation of this by PKA, protein kinase A, results in a profound increase in pain memory, right? So with opioids, what we're doing is we're inhibiting Adenolith Cyclase. So we're preventing the conversion of ATP into cyclic AMP. So we're, stopping or I don't know, like eliminating we're decimating our army, killing 1 out of every 10. That's what of our own. That's what decimation means. We all know what a decathlon is, right? 10 events, whatever, deciliter or whatever. So decimation, you kill literally 1 out of 10 of your own. So we're sort of decimating our cyclic AMP. And so that doesn't activate as much PKA, so we don't get as much of this CREB signaling. So we don't get as much pain, but also there's a lot of other functions that it's going to have. And learning is going to be one of them. So modulate pain and long-term memory and other physiological and pathological processes. So regulation becomes so important. And we have our endogenous opiates, right? We have, you're going to go run, and like anandamide, right? And would be an endogenous cannabinoid, but the endogenous morphine that we have, like that's when we're looking at all these effects that are giving us some pleasing, I guess anandamide would be like the runner's high mostly, but like endorphins people talk about as the runner's high. There's going to be a little bit of that. That's endogenous morphine. Endorphin, endogenous morphine is what it stands for. And all of these things, some of that effect, a lot of that effect with endorphins, and certainly a lot of that effect if you're putting in some opioids, are going to be through this of halting that Pka signaling cascade, but you're going to get loopy at the same time, right? You're going to impair learning of more than just pain when you're doing this stuff. So there are consequences. Getting into the cannabinoid receptors, it was the first one identified, this little pathway, where you see Cb1 and Cb2, that cannabinoid receptor 1, cannabinoid receptor 2. And this is what you're in your weed. There's 60, active compounds in that, but the most psychoactive, the thing that kind of gives you the psychoactive effects is going to be your THC. And so we have the Cb1 receptor, the Cb2 receptor, and the first ever identified pathway where those work through is this Pka signal. But it turns out there's a lot more. And maybe there's a Cb3 receptor, um, a forthcoming identification of a Cb3 receptor, because there's so many other interactions that we're seeing. And so over here, if you just look at the table of contents of this one, we see the G protein coupling. Okay, down here, regulation of Denylol cyclase. So adenolith or Adenylol cyclase, that's the exact same thing. And again, it's like MAPK, mitogen activated protein kinase, the ion channels, the calcium and opioid receptors as well. There's interactions that they have with opioid receptors. There's just so much stuff that the cannabinoids are going to be doing. And again, the first thing that was ever identified, and what people pay most attention to is that Pka signaling, which is the same thing the opioids are doing. It's having a similar effect of opioids. Now, prostaglandins, you would see over here somewhere, prostaglandins would be initiating this Pka signaling. They'd be turning it on, that Pka, and inhibition of prostaglandins by taking an NSAID, by taking a non-steroidal anti-inflammatory drug like an aspirin or an ibuprofen or something, taking one of these, you're not going to have as pronounced effect by inhibiting prostaglandins and then by way of domino inhibiting the activation of Pka. That won't be as pronounced effect as if you take an opioid, right? Take an opioid to knock that thing out, or cannabinoid to knock that thing out. But again, there's a bunch of these different functions of the cannabinoids. And yes, we have that Pka signal, and that's what people talk about the most, and that was the first thing identified. But there's a great deal more diversity, and here's the tables from it. And this isn't that new of an article, right? 2006. This is pretty old, and there was still so many pathways already identified, and we haven't made that much progress in understanding cannabinoids, because early in Wednesday's lecture, I mentioned that CBD and THC are both still lumped into this same, although THC is a psychoactive compound, they're both still lumped into the same class. Federally, all 50 states have some sort of permission or accessibility. California is sort of fine for getting the cannabis. And it's sort of silly why some of these drugs is silly. It's a little bit more nefarious than silly, why a lot of these drugs were illegal in the first place. Because you saw that list of, like, alcohol is the worst one, and then heroin, and then we're getting to like crack and meth and whatever, and then tobacco, and you get pretty far off to the right, to the low end, and you're seeing totally illegal stuff. but there's a lot of legal stuff at the top, and there's more to the story than this. But if you just think about the races that the ethnicities of people who did these drugs, people for whom this was culture. So what's like the safest of all of these drugs? Well, like peyote, shrooms. Like just nobody's ever had like a problem with shrooms. Totally illegal. Well, who was doing that? Well, I don't know, Native Americans. And like, okay, let's make that shit illegal then. Um, African Americans was more cocaine. Um, when we get into, oh, I don't know, what other drugs are the opium and we're looking at more Asian there's all we can just identify a population that's not just a bunch of like white people, and we made all those drugs illegal. What were the drugs of just like whiteys? Is well, just like alcohol and cigarettes. Okay, let's make sure those ones are fine. Now, again, there's more to the story, so one shouldn't get carried away with a narrative, but the narrative is compelling, and it has nothing to do. These illegalities have nothing to do with safety. They have nothing to do with consequences. Commonly, they have nothing to do with addiction. It's just politics is often what it comes down to. Um, anyway, so cannabinoids, people have for a long time had this anti-cannabinoid position, and and even the NIH, the National Institutes of Health, prevented funding for people. If you were exploring this, good luck getting funding in the future. I mean, there are these prohibitions here. And so we see some shoddy work, we see limitations of work, and it looks like a lot of studies have been done, but it's not really true. I mean, look at any drug, and it's just so much more studied than cannabinoids, so much more understood than cannabinoids. And cannabinoids are one of the most complicated drugs that exist in the world. The number of signaling cascades these things work through, the number of interactions that they have. Again, we see this, the regulation of adenylol or Adenolith Cyclase, but there's tons of other things. Now, this stuff, going through stuff that's not Cb1, Cb2, this stuff going through those cannabinoid receptors. And arachidonic acid, we're seeing this over here, increase of arachidonic acid. You know what that is. You know what NMDA receptor, you know what those are. There's your PI3K, PKB. We see a plus, but that's conditional. That one depends. MAPK, this is all mitogen activated protein kinase. That's a parallel road to mTOR, to PI3K, PKB, mTOR, mitogen activated protein kinase, or ERK, RSK, stuff like that. So their concluding remarks in their article is just, there's Cb1, Cb2, these do a lot of things, but there may be more. There may be more types of cannabinoid receptors than Cb1, Cb2. Still today, people say, well, potentially there could be a Cb3. How about this is a candidate for a Cb3? People have stories that they will kind of, some articles will still talk about the Cb3 opportunity for nomenclature for this particular pathway or that particular pathway, but there's more to it than just Cb1, Cb2. And even in these receptors, it's just so diverse. So what they conclude is just it's so complex. Cannabinoids are so complex. Now, where it's simple, where cannabinoids are simple and everyone gets it and everyone has understood this for quite a while. Now, it may be more complicated than this, but the rudimentary story that has withstood the test of time and the scrutiny of science is hypothalamus regulates our food, right? If you want the munchies, THC is going to have you in the kitchen. I mean, THC, you are going to find yourself in the kitchen. There is a drug, Rimonabant, I think it's pronounced, that's a Cb1 inhibitor. It's an inverse agonist, a Cb1 inhibitor, and it's an appetite suppressant. Right, that thing is an appetite suppressant. It's not legal for humans. It was never legalized in the U.S., and in Europe, it was legalized for 2 years. And then, oh shit, people get terrible psychiatric conditions when they're on this. So I think in 2008, somewhere around there, it was pulled off the market after about 2 years of being on the market. But a CB1 inhibitor is an appetite suppressant. And where is our appetite controlled? Well, up in the hypothalamus, that little brain hunk. Up in the hypothalamus, cannabinoids are going to turn on AMPK. It's the opposite effect that leptin's going to have. Insulin too. Insulin's a bit like leptin, but it's the opposite effect that leptin will have. So leptin is going to suppress appetite. Cannabinoids are going to have us in the kitchen munching. And if we turn on AMPK, remember, AMPK is an energy sensor in the body. If AMPK is active, AMP activated protein kinase, if that kinase is active, we're trying to increase our ATP levels. If AMPK is inactive, we get to spend money. We get to spend our ATP. And so AMPK gets turned on. Let's say it's by like resveratrol or something, or just exercise, you get a bunch of AMP, right? Then LKB1 is going to turn the thing on. And in the hypothalamus, we're going to eat food because that's how we're going to get the sustenance ready, those macronutrients ready to reconstitute our ATP. In the periphery, we're burning those calories, right? We are burning up our carbs. We're burning up some fat. We're trying to reconstitute ATP levels. So you will see a little bit of in the periphery as compared to in the hypothalamus. There may be some what appears to be contradictions or counterintuitive arrows, but it all makes sense if you just know what AMPK does. But cannabinoids, AMPK in the hypothalamus, munchies. That's fine. We all get that. By get it, I mean we understand the concept, the principle. It makes sense. Nobody's arguing AMPK in the hypothalamus with cannabinoids. But when you get into mTOR, when you get into the foundation of anabolism and catabolism and the balance of the buildup and degradation and this sort of critical stability point of all of our metabolism, well, we don't really we don't really know much. We're not very good at that part of it. So we'll go through a couple of studies on, well, we'll cover the same couple of studies on CBD. We'll just go through them one more time, and then we'll do the same thing with THC. We'll go through 2 studies, and obviously, you know, I'm gonna show you 2 studies that just oppose each other and say the opposite thing, because that's the state of evidence at this point. So you remember this, those Ncaa cells, immortal cell line. They'll just kind of go and go and go and divide and divide and divide without, you know, cessation. But so the CBD cannabidiol is deactivating the mTOR pathway in this one. Now we are in T cells, right? So we're in an immune cell here. And but what they found was it's dose dependent. You got to get it up there a little bit. And phosphorylation of PKB, remember AKT is PKB. That's the exact same thing. This isn't like a ratio of something. This is just PKB, AKA AKT. They're just identical. It's 2 different names for the same thing. And in the presence of cannabinoids, we are reducing over here. So deactivating the mTOR pathway by reducing the activity of PKB. Now, PKB is going to phosphorylate tuberin. Remember, tuberin is deactivating Reb, preventing Reb from turning on mTOR. So downstream from mTOR and downstream from MAPK, phosphorylation of ribosomal protein S6. Remember, translation happens at a ribosome, and so a reduction in that phosphorylation, a reduction in the upstream kinase of PKB. And the time course of this, right, here's 0 hours, here's 8 hours. Just look at the ribosomal protein S6. Just starts to taper, right? Couple hours in, 4 hours in, 8 hours in, we are limiting, we're reducing the phosphorylation of ribosomal protein S6, and you see the same thing with phosphorylation of the mTOR. There's the mTOR enzyme itself. but it's nice to look at that ribosomal protein S6, because that's really the last stop before you're translating some protein. So there's a time component and there's a dose component to doing that. But yeah, so CBD would suggest the PKB-mTOR1 route, the deactivation of it by application of CBD is the deactivation of mTOR. Going to this one, going to this article, is they're finding the increase in mTOR signaling using CBD, finding the increase in mTOR signaling in the medial prefrontal cortex and the hippocampus by way of BDNF, brain-derived neurotrophic factor. Remember, that one binds to TrkB. So there was TrkA, nerve growth factor. That protein binds to TrkB is BDNF, brain-derived neurotrophic factor. And again, in the hippocampus and the medial prefrontal cortex, that is where we're seeing activation of mTOR going through that BDNF signaling cascade. So BDNF binds to TrkB, and downstream from that, we're getting mTOR activation. And if you put in, so rapamycin, mammalian target or mechanistic target of rapamycin, that's what mTOR stands for. It's a target of the drug, of the exogenous drug. This isn't an endogenous thing. This is an exogenous compound. Mammalian target of this thing. So this shuts down mTOR like fierce. It interacts with the mTOR complex itself. It's not an upstream inhibitor, like something like wartmannin would be a PI3K inhibitor. And so you're knocking out a lot of mTOR, but not the whole thing, because you can still get in there through mechanical loading. This just shuts down mTOR. So even mechanical loading, you introduce this and mTOR is just done. So they administer the rapamycin, and the CBD administration, you're going to knock out that BDNF signaling at the same time. Yeah, it's just like same stuff, TrkB activation, BDNF, whatever. Okay, so we've done CBD, and the state of evidence is a little bit messy because it depends where you are. It depends on contexts, and those two studies say, one of them says mTOR is shut down. The other one says mTOR is turned on. Okay. Here's your THC. We'll talk about that in just a second. But these are our exogenous cannabinoids, right? Here are a couple of endogenous cannabinoids. And these are going to work similarly. You could take these as a drug and they'll work similarly. They'll be a little bit different, but they will work similarly to cannabis. And one thing that the cannabinoids will be doing is augmenting the release of opioids. And so you will get this, I don't know, that sort of light. If you've done opioids before, you've been at the hospital or something, or you got a prescription or whatever, or recreationally, whatever. You know the effect that they have, that there's a little bit of overlap in that sensation that you can get from cannabinoids. And in part, that's going to be from the upregulation, from the increase of enkephalins and these endogenous opioids that you'll release, dynorphins, and there are specific opiates or opioids that you'll release in response to your cannabinoids, and they have a similar sensation. But let's talk about THC for a minute. We'll do two articles. So with this one, the Cb1 Cb2 receptors, it's just a nice little explanation of what it's doing. And we're seeing a lot of this. Cb1 is central nervous system, really abundant in the central nervous system. It exists outside of the central nervous system. The Cb1 receptor exists outside of the central nervous system, but that's really prominent in your CNS. The Cb2 receptor. That one is mostly immune system, and does it exist outside, like elsewhere? Yeah, sure, but most of it is going to be kind of peripheral immune, and CB1, most of that is going to be central nervous system, and um, so they had it's a bunch of like mice in all of these studies, and the memory. So they were looking at the THC on the effect of cognition and memory and chronic THC administration. One of the interesting things is that we see it depends on the drug. It depends, when we're talking about specificity of adaptation, it depends on what we're exposing ourselves to. But down regulation with chronic THC administration. So with a lot of things, if you exercise and get more androgens going, something like that, you're going to get an upregulation of androgen receptors. Now, behaviorally, endocrinologically, these things are going to initiate an upregulation of androgen receptors. So you have enough receptors to bind. But with something like THC, if you administer it chronically, you are overriding a system, and that system tries to correct, right? Tries to find balance. And so you're getting this pronounced effect at a given dose. And the tolerance that we develop in part is, remember, the Cb1 receptors, that's the one that's most concentrated in the central nervous system. Those, you'll see a downregulation of the receptors in response to excesses of use, so that the use doesn't have quite as profound, as pronounced of an effect. Although the cognitive impairing effects do seem to linger a little bit, but this article does seem to carry, I'm not saying it does, but it seems to carry a little bit of anti-THC bias in it. So the administration of THC here, they found activation of MAPK, that's mitogen activated protein kinase, right, or extracellular signal regulated kinase. We think of these as the same cascade. They end up with RSK, very, very anabolic. This stuff is very anabolic. And also PI3K, and then AKT, remember that's PKB, same thing. And so this is mTOR. So over here, and these two roads converge. These two roads converge as well as intersect, so that they end at the same place. They have intersections earlier on, but we see THC activating both the MAPK cascade and the mTOR signaling cascade. And what they say, like, interestingly, because it is a little bit of a paradox, right? The mTOR pathway activation was involved in memory impairment here, produced by the endocannabinoids, right? The THC. They give people THC, and if they give them rapamycin, it blocks some of this, the sort of like rapid fire amnesia people get when they're on THC, that you remember stuff for like 1 second. You better write it down or record it or something, because that thought is going away in 1 second if you don't document it somewhere. If you take rapamycin, mammalian target of rapamycin, that major mTOR inhibitor, like the most powerful mTOR inhibitor we have, if you take that, you block some of this amnesia effect. So some of the psychological effects of THC are routed through mTOR signaling. Now, they sort of make this assumption that that's kind of like a bad thing in general. But if you look at, how does a brain grow and maintain itself, and nerve health, and your dendrite formation and all that, this is mTOR. mTOR does that. So if you turn on mTOR in your brain, we're looking at a lot of, just piles of data saying that's a good thing unless it goes too far. If you have chronic dysregulation of mTOR, tuberous sclerosis complex, TSC, and TSC2 is tuberin. Tuberous sclerosis is a condition. That's a condition. You're getting all these tumors growing in your brain, these benign tumors, just out of control growth. That's not good, right? So if you have disruption of mTOR, where you have this suppression of tuberin activity, you're going to get this constant activation of mTOR. That's not good. What you want is regulation. You want balance. You want mTOR to be firing appropriately. But when we're getting nerve, not necessarily regrowth gets into some touchy territory, but in the central nervous system. But I mean, look up like Fred Gage's lab. I mean, there's a lot of data on that, but that's not what we're talking about. But when we are trying to preserve the health of our nerves and so neurogenesis and neural communication and dendritic sprouting and all this, it's mTOR. mTOR is doing that. So shutting off mTOR in the brain would be a bad idea. And when they say like turning on mTOR in the brain is what's going to like wipe your memory and stuff, that's maybe a little bit misleading. So they have these Cb1 knockout, Cb1 receptor, that's what they are, is knockout mice, and then they have these wild type mice. THC as compared to vehicle, as compared to nothing, as compared to like inert, like what would the THC would be in, but the THC is not in. So this is your control group over here. And these are just different brain areas. So hippocampus, striatum, cerebellum, right? These are the different brain areas over here. And then looking at phosphorylation of P70S6K. And, in the THC group, we're seeing more phosphorylation of P70S6K in some of these brain areas. So THC, let's turn on mTOR. Remember, P70S6K, that's the kinase that's immediately downstream from mTOR. mTOR phosphorylates that thing. If mTOR is on, and Raptor is working, Raptor is recruiting as downstream targets. If mTOR is on, it's phosphorylating P70S6K. So this is evidence of mTOR phosphorylating its downstream target. More phosphorylation of these things in several brain areas in the presence of THC as compared to the control, as compared to the people receiving like inert, fluid. All right, so let's go to a new study. And so this is mouse pancreas, and they're looking at the relationship between Cb1 receptor, again, that's the one in, it's in the periphery, but it's, concentrated in the central nervous system, but it is in the periphery as well. And mTOR. And so, P70S6K, mTOR, P70S6K. This right here is that drug I mentioned earlier that is the inverse agonist of the Cb1 receptor that's like totally illegal for humans. You can't use it for humans. Like a doctor can't prescribe that thing. There's just too many psychiatric side effects to it. But if you introduce a Cb1 receptor antagonist, a blocker, then you can see what the effects would be, and it would be a wonderful appetite suppressant. But so what they found, they introduce this Cb1 blocker, and they're getting an increase in mTOR signaling, in mTOR signaling in the pancreas with a Cb1 blocker. So without that blocker, they wouldn't be getting the increase in mTOR signaling. So either the last article said you activate the Cb1 receptor, right, with THC, you activate the Cb1 receptor and you get an increase in mTOR signaling. This article says you block it and you get an increase in mTOR signaling. Over here, we see opposing effects. So rapamycin, that's mTOR specific inhibitor. So you see it down here, rapamycin works with this thing called FKBP12 in the complex. But they did both rapamycin and rimonabant, or however you pronounce this thing. And so they inhibit the Cb1 receptor and they inhibit mTOR, and it doesn't change anything with the P70S6K phosphorylation or the phosphorylation of ribosomal protein S6. So we have two opposing positions on CBD. right? We have two opposing positions on THC in terms of the metabolism of it, in terms of mTOR activity. What is happening with anabolism, whether you're in a nerve or you're in your pancreas or you're in muscle or whatever you're in? We're just seeing these opposing effects. So I don't really have a consensus. I don't have a position statement that I can give you on any of this stuff. But what seems most likely is that, like, it's there doesn't seem to be harm. There does seem to be, if you disrupt mTOR, you get neurodevelopmental problems. If you turn on mTOR too much, if mTOR is really, the engine just won't shut off, you're going to have tumor issues. And so on both sides, you want regulation, but like acute doses of THC or CBD, we don't really have any good information on it, other than it does seem to have an effect on mTOR, and that seems to be circumstantial and regional. What cell type are we in? What conditions? What kind of creature? So I don't know. That's my unsatisfying end to cannabis. Do we have any questions? Are we all just like, Friday is here? I am so ready for Friday. It's been such an exhausting week. Is that like, the chat box is open. Nobody was typing. Do you guys, like, is this just the most boring subject in the world? Or are you like, it's Friday at this point in the semester?
Yes.
Super complex. So is cannabis good or bad? Yeah, I don't know. I think the goal is that you can use cannabis effectively and it's unlikely to cause harm. It can be overdone just like broccoli can. You don't want too much sulforaphane. It can be overdone just like anything. And the idea of being, quote, stoned, that's not like a productive place to be, you know? Uh, there's a lot, yeah. But you can get advantages out of it. If, like, let's say they for seizures, THC seems to be bad for seizures. THC does. CBD seems to be about as good as it gets for seizure treatments. For pain management, CBD seems to be, some of the difficult inflammatory conditions, some of the difficult neuropathic pain conditions. CBD seems to be about as good as it gets for treatment without having too much of the, cognitive disability that comes with something like gabapentin, that this sort of acute, like, let's just stay fine. No matter where you are, if you've ever taken gabapentin, I can say this from experience, no matter where you are, you can find an abyss to just stare into and like practically drool. That's gabapentin, right? CBD, that doesn't happen to you. It's possible you get a little bit sleepy or something, but you don't get that. And you may even, that article I showed you for in the brain, get the BDNF, right? So BDNF, there's like the best brain health you can ever get. You're not going to get a bunch of BDNF from gabapentin. So should cannabis be banned by the Ncaa? I don't understand, like, the bannings of stuff. I always find a little bit weird. Like, what's it gonna? It would help with target shooting or something. But in Mike Tyson's memoir, The Undisputed Truth or whatever it is, he talks about, because you can watch all the. That's the nice part about the 21st century is you can just go to YouTube and like watch every professional fight that he ever did. And he talks in his books about what drugs he's on in the fights. And it's like, look how slow I am here. Here's the drugs. And it's not helpful. THC and CBD are not helpful for that. It's really helpful if you're like a musician. You can tell Neil Young. He's in the Rock and Roll Hall of Fame twice. He was inducted twice, once as a solo act and once as a band. He wrote his memoir called Waging Heavy Peace, I think it was called. It wasn't that good, but Neil Young is whatever. He's one of my guys. So I read his book, and he hasn't put out a song in a long time. He said, like, yeah, the last time I wrote a song was the last time I smoked weed. It's just like every time he did cannabis, he wrote a song. If the dose is right. If the dose is too big, he couldn't write a song anymore because he was stoned, which is just not helpful. That's just like you're boring and stupid. But if you use a certain dose, you just hit this like creative space. And it was interesting listening to Neil Young say, like, yeah, the last time I wrote a song was the last time I smoked weed. I gave up weed and I haven't written a song since. And that's, I think I think musicians might not say that. For recovery, it would be an interesting series of studies to run. Like, what's going to happen? Like, yeah, okay, Michael Phelps made it famous in, like, Olympic sports, but I don't know, he was just like being a kid, you know. I don't know that he was using it in a calculated way. Is it possible to use it in a calculated way? Maybe. It's not really a PED, yeah, performance enhancing drug. Not really. Yeah, it just depends on. It's a performance enhancing drug for creativity, and maybe if your sport is something it's like your chess, playing chess, or like poker or something. Like if that can be counted as a sport, that I would argue they can't. But then maybe there's some sort of creative strategy you can come up with, this sort of lateral thinking that you would have not bounded so far in the lateral direction if you were forced to think with a sober brain. Anything conditionally can be PED. It just depends on the conditions. But yeah, it's not going to help you be like a baseball player, a water polo player. I don't know. You do focus on reps. An individual rep is like something that gives you great attention. Like, wow, how fascinating is this bicep curl? And it's like, it's weird. And so you end up doing these like great sets in the weight room. Like, I don't know, like obviously I've worked out and on THC before. And I've never had such a good set of like tricep press downs. It was somehow the most meaningful thing in the world. And you do like 1,000 perfect reps, and it's just fascinating the whole time. And then you're sore for like 3 weeks. Yeah, try it. Seriously, you'll never have a better workout. You have to, the space has to be right. Just like with alcohol, you have to be in that magic zone. I think with THC, the magic zone lasts for so long, and you can overshoot it just like you can with alcohol, but if you undershoot it a little bit, you still get some of that fat. You have to be in the magic zone though, but the magic zone lasts for, longer than a workout does. But yeah, interesting workouts. Yeah, I mean, you'll be sore for like 10 days afterward. All right, I'll see you, JP.
Is there any differences between like ingesting it like as an edible compared to smoking it on the body?
Yeah. So, like, but can we just have like a THC talk for a second that has like that's like we're just recreational. So like the tinctures, you can really control the actual strain and the dose and everything with the tinctures. Like the little bottles that like you can actually get your dose in the edible and like the gummies. I mean, the funny part is with the gummies, I think people probably overdo the gummies a little bit because they get like the munchies, and then they have gummies, and they're like excited about the food part of it. And so I think people might, but it lasts a lot longer. It seems to set in slower, and depending on the dose, there seems to be this very prolonged kind of coasting. Um, and so the edibles do seem to last a little bit longer. I've like I've only smoked once because I don't really like. So you don't control the dose, and in the actual, like marijuana itself, it's notorious for having aspergillus in it. So if you smoke, you're just inhaling mold, like super toxic stuff. I mean, it's just notorious for like people get mold toxicity off of that. And having had really severe case of mold toxicity, like I don't want to smoke. But yeah, controlling the dose and the strain and everything, tinctures seem to be the best way of doing it. Because also, Yes, indica and sativa. There's these different. There's kinds that just make you depressed. There's kinds that make you giggle. And so I'm not a pro when it comes to the different strains of it. And like the best strains uh, have names that like you're embarrassed to buy, like green crack. Like I don't want to buy a product called green crack. It's like it's the best one, but I don't want to buy that. Any any other questions about any of this? All right, should we go do weekends?
I actually have a comp
Yeah.
question.
Okay.
So I was gonna go after if like anybody had like more questions.
I had a scheduling question too regarding classes.
Okay, I'm going to shut off the recording and stay on, and we'll just do like if you have comp or advising questions or whatever, we'll just start tackling those.
Okay.